Hi, I'm Stacy Montgomery, and thank you for joining us on this webinar on using metabolic analysis to optimize bioprocess conditions. Two important aspects of optimization are the development, selection, and characterization of production strains, and how to develop optimal growth conditions to produce the highest yield possible. In the next few slides, I'll show you how the technology works to provide a comprehensive global picture of your production cell lines and how you can use this technology in different stages of development. There are many ways to take a comprehensive or global look at cellular properties. The issue is that these transcriptomic or proteomic approaches often show many changes at the transcription or translation level, but there may be downstream modifications or processes that result in no discernible effect on the cell. So this means there's an enormous amount of data to sift through to find what is actually meaningful and actionable. So while they result in massive amounts of data, it can be difficult to determine how those changes actually affect a cell. So we've taken a different approach and instead focused on cellular phenotyping using a universal respiration reporter that we can link to many different compounds or conditions to get a functional metabolic readout. So we have a series of redox reporter dyes that allow us to link cellular respiration to our compounds of interest or different conditions. If the organism can utilize the compound or respire in the presence of components in the well, they respire, they produce NADH, and that NADH can then be used to convert our colorless tetrazolium dye into a visible purple color. So by looking at expressed cellular properties or phenotypes, we're eliminating much of the noise and can focus on properties that are biologically relevant to the cells of interest. The platform for all our assays is a standard 96 well microtiter plate. Each well contains a different substrate or an inhibitor along with our universal respiration reporter, and if the organisms can respire under this condition, the well turns a visible purple color that's non-reversible under physiological conditions. For bacterial cells, we have a series of 20 plates. The top 10 plates are focused on metabolic conditions, where we look at carbon metabolism, nitrogen metabolism, phosphorus, sulfur, osmotic and ionic effects, pH effects, and then the second set of plates at the bottom are chemical sensitivity assays. The easiest way to think about what we're doing is that each well is testing for the presence or absence of a known biochemical pathway. These are not random chemicals that we used because they were inexpensive, stable, easy to source. They were all designed to interrogate a specific biochemical pathway known to be present in bacterial cells or a particular condition such as optimal pH, ability to withstand salt, etc. The same thing is true for the inhibitor panels. We have a wide range of inhibitors with different mechanisms of action. So it's like we're poking the cell in different places to see how it responds to all those different insults. These assays are very simple to run and consist of the Phenotype MicroArray plates, the Biolog incubator reader, and software that's designed to interpret the results. So this is an example of a typical experiment where we're inoculating two plates with different cell types. So you can think of it as a wild type versus a mutant strain, or it could be untreated cells versus cells treated with a particular compound we're interested in. But we inoculate our plates, one type labeled in red, another type labeled in green, place it into the Biolog system. It takes a time point every 15 minutes, and then we get the plates with the peaks showing the phenotypic activity. The software then allows you to overlay those particular curves for the different cell types, and everywhere you see yellow, the cells are the same. If you see a red peak or a green peak, it means there's been a change in phenotype between the two strains that you're comparing. As I mentioned, these are kinetic curves for each well we're looking at, and we're looking at respiration over time. And then we can investigate the lag, the slope, or the maximum response for each substrate or test condition. This technology can be used for two important aspects of bioprocess development: strain development and selection of clones, as well as optimization of culture conditions for best yield. And we'll look at those two aspects of the technology. The typical methods for process development are slow and inefficient. So on the left side of the screen, we typically start with a production strain, modify that strain, creating daughter cell lines with better production traits. We then choose the clones that we're interested in and manipulate the growth conditions such as the media, the pH, different conditions, and then we screen a large number of those clones for increased yield and titer. So this is this iterative circle where we start with the strain, mutagenize it, select clones, measure their titer, then we grow under multiple feed conditions, measure the titer again, and if you get lucky, we proceed to scale up. If not lucky, we start over. So this is a process that oftentimes gets repeated over and over again. So let's review a few different kinds of experiments that can be used to analyze or compare your strains. This first one is a typical phenotype microarray comparison of two strains. It's a wild type MG1655 E. coli strain and a mutant that we've developed by transposon mutagenesis. In this case, we've inserted TN10, which carries a tetracycline resistance gene, and we've inserted that into the MALF gene. And what we see when we compare the wild type to the mutant in this experiment is we see that we've lost the ability to utilize maltose sugars as we expected. And we can also see that we gained resistance to tetracycline and confirm our genetic construct. Another thing we can see by looking at this is we can see that we didn't have any off-target effects. This is a more interesting example of mutating a regulatory gene. In this case, we've interrupted the OxyR gene by inserting a kanamycin-resistant cassette. So the OxyR gene that we've mutated here is a global transcriptional regulator of the oxidative stress response. And we can see that this mutation affects a variety of cellular phenotypes. And this is a very interesting example of being able to globally or comprehensively screen your cells. If you're dealing with a regulatory gene, sometimes it's hard to predict what other genes this is affecting. So in this way, we can see that we've mutated our gene, our OxyR gene. We can see that we've introduced aminoglycoside resistance, and we can see a whole range of changes as a result of this global transcriptional regulator. Sometimes there's a need to discern the differences between two closely related strains. In this case, we're comparing two strains of the same species, a pathogenic E. coli 0157 as compared to MG1655. And people have understood that 0157 behaves differently from typical lab strains of E. coli for a long time. In this example, everywhere you see a black box, the software is flagging a significant difference between these two strains. So experiments like this can be really useful to determine changes in strains due to phenotypic drift. For example, if something feels off about your production strain, you can use these types of experiments to detect what those changes might be. And then later, you can also use this as a very comprehensive QC test to ensure the strain that you're working with today that you'll be using behaves exactly like the strain that you were using 2 years ago. So how do we correlate these color changes in wells to the metabolic function and figure out exactly what's going on with our strains? So let's take a look at the Omnilog software and see what the options are. So the Omnilog PM software is linked to the Kyoto Encyclopedia of Genes and Genomes, or the KEGG database. And so we can click on a single well on a plate, and the software will tell us what the compound is in the well, along with the mechanism of action. In this case, we can see I've clicked on a well A3 in PM plate 1, and I can see that this is N-acetyl D-glucosamine, and it's a carbon source. One more click, and it will show me the molecular structure of that compound. Another click, and I can get to the enzymatic conversion and the products of that conversion. Another click, and we can get to the enzyme that catalyzes the conversion. And another click, we've got the amino acid sequence and the nucleotide sequence of the gene for that enzyme. So within moments, we can get to this end result, which is a pathway analysis at our fingertips. So to summarize, we looked at the technology and how it can help us develop, select, and characterize clones with useful properties. Now we can look at how it can also be used to determine optimal conditions for growth of our cell line. So once we've selected our specific clones, it's time to optimize growth conditions. We can now take our prospective cell lines and determine the optimal carbon source, nitrogen source, pH range just by inoculating the proper plates and incubating overnight. This is much more efficient than performing shake flask studies for each individual compound or condition on multiple clones. So in this way, in an overnight experiment, we could actually test 1,920 conditions for each of the individual clones. The goal is to find optimal culture conditions as early as possible. And this is some work that was shared by Nigel Mounsey when he was at Dow AgriScience. And his goal was to determine optimal conditions for the development of a new product. So he was looking for carbon sources that supported growth. And in addition to measuring carbon source utilization, he also tested the titer of the product in each well. Those wells that demonstrated a decrease in titer were labeled in green, and those that had an increased titer were labeled as purple. And what he found was culture conditions that support the best growth don't always result in the highest yield of product. So we need to find the optimal carbon source and conditions for growth, but also for optimal yield. In addition, while we're doing this, we can look at alternative carbon sources that may be less expensive, and once we get to scale up, this could end up saving us millions of dollars in production costs. Now we'll take a look at an experiment where a client wanted us to examine their production strain along with several daughter cell lines and provide input on optimal growth conditions. So we ran their parental strain in our lab and 3 of their daughter cell lines on the PM or Phenotype MicroArray metabolic panels 1 through 10. So when we tested the parental strain and looked at the baseline metabolic response, what we saw was a little bit surprising because it was pretty non-reactive. If you look at the PM panels labeled 3, 4, 5, 6, 7, and 8, you can see that there weren't a lot of positive reactions. What we found out upon reviewing the data was that the client's production strain actually required leucine for growth. This need was unknown to the client, but made sense based on the formulation of media that had been shown to be reactive with this strain. So they were dealing with a leucine auxotroph. The client wanted us to continue testing the daughter cell lines to find out more metabolic information if we could. So we found that all the daughter cell lines, when we compared them, showed increased sensitivity to salt. So we exported the data and compared it side by side in a graphical format. And you can see easily that the daughter cell lines had become more sensitive to salt than the parental strain. So our observations for the client were that the parental strain and daughter cell lines were all leucine auxotrophs, and all 3 daughter cell lines were more sensitive to sodium chloride concentrations. So they need to include leucine in the production media and manage their salt concentrations. The examples I've shown you so far have been in bacterial production strains, but the technology also works for yeast and filamentous fungi. And this is an example of how a single microplate was used to look at carbon source utilization, secondary metabolite production, antimicrobial properties, and they actually used it to dereplicate their culture collection. So this is work that was done by Maya Singh at Wyeth a number of years ago using a single FF microplate that was originally designed to identify fungi to the genus and species level. What he did with the plate, he took his culture, they don't tell us what it is in the paper, they don't tell us what they're trying to produce, but you can guess from these experiments that he's looking for production of a secondary metabolite that inhibits Candida albicans. So what he did was take his strain of interest, inoculate the FF microplate, and incubate it. Then he read it on a standard plate reader to see which carbon sources in that plate supported growth of his organism. For those substrates that supported growth, he then scaled up into 1 liter shake flasks and measured the target metabolite. He also took the supernatants from the FF microplate and then used those to test against a lawn of Candida albicans to see if there was antimicrobial activity in the supernatant. What he found was that gentabios and D-fructose supported the highest levels of growth. But on his scatter plot where he was measuring the compound of interest in the shake flasks with the substrates that were best for growth, the highest result was obtained with sucrose and L-rhamnose, not fructose or gentabios. And when he tested those supernatants from the FF microplate for antimicrobial activity with Candida albicans, sucrose and rhamnose were the substrates that led to the highest inhibition. So we can see from these experiments that the preferred carbon source can be decoupled from the carbon source that will allow for the highest yield or titer of our product of interest. So I've shown you how the technology can be used for all types of microbial cells for clone selection, characterization, and determining optimal growth conditions. I also want to mention that we have the same technology optimized for mammalian cells as well. So it's the same basic technology, but the compounds and protocols were optimized for interrogating mammalian cell lines. I covered a couple of use cases on strain development and characterization, as well as optimization of culture conditions leading to higher yields, but we've also seen our technology used in novel ways for a lot of other applications. So if you have any questions, please feel free to contact us at info@biolog.com, and I would encourage you to go to our publication database, which is searchable. I've got the link right here, so you can search the database with country of interest, product of interest, organism of interest. So take a look at the database and please email us if you have any questions.